Hello everyone, my name is Cyrella and I'm a student at MHHS, and today I'm going to be presenting to you about my Landoltia punctata cDNA clone. So Landoltia punctata is commonly known as duckweed, and it's a freshwater aquatic plant that is of interest to the biotech field. In this research project, I've analyzed DNA sequence of genes from this plant and determined which protein it codes for. The first step of this project was to crop the ends of the DNA sequence so that it is separated from the vector, and then to edit the actual sequence so that there are no missing or additional bases, peaks that have not been determined that are marked as N, and bases that were wrongly identified. The next step was to perform a BLAST-N search in order to determine if my DNA sequence matches any other DNA sequences that have already been determined and been deposited in the databases. The BLAST-N search compares a nucleotide query sequence against a nucleotide sequence database. After performing a BLAST-N search, I performed a BLAST-X search. The goal of the BLAST-X search was to determine if my DNA sequence likely codes for a protein. Instead of comparing a nucleotide query sequence against a nucleotide sequence database, a BLAST-X search compares a translated sequence in all reading frames against a protein sequence database. The fourth step was to determine the open reading frame, aka the region that codes for a protein. After determining the open reading frame using Toolbox, I used the ORF to run a BLAST-P search, which, like the BLAST-X search, compares the entry against a protein sequence database, except it's using a protein sequence instead of a translated DNA sequence as the query sequence. After performing both the BLAST-P and the BLAST-X searches, I compared the two results to make sure the open reading frame was correctly identified, and then determined where the 5' UTR and 3' UTR were. The last step was to analyze the protein found by my BLAST-P and BLAST-X searches. The protein I found, as you can tell by the title, is the proline-rich protein. So in my bioinformatic analysis, you can see that my BLAST-N result showed a very similar nucleotide sequence, and my BLAST-X result showed the proline-rich protein. So in mammals, proline-rich proteins are salivary proteins, but in plants, they perform a different function, and they make up a huge part of cell wall proteins, which play important roles in cell wall signal transduction cascades, plant development, and stress tolerance. When I search PDB to determine if there is a homologous protein in which the structure has been determined, there were no results, because proline-rich proteins don't have a fixed shape or organized three-dimensional structure. They are called intrinsically disordered proteins, or IDPs. I also found that they are highly localized in the cell wall and less in the nucleus and cytoplasm, which makes sense because in plants, they make up a large part of the cell wall proteins. And they're also highly expressed after many hours in experiments involving needles or tubes. This is probably because they puncture the cell wall. And as I said, again, they play a large part in cell wall proteins. So on the right, you can see my results, and you can see that they are highly expressed, highly localized in the cell wall, and also in the nucleus and cytoplasm. And you can see on the right also that after punctuation of the leaves by 3 consecutive applications of pin tool with 16 needles, they're highly expressed in the leaves. So after completing this analysis, I'm very curious about how the protein behaves in mammals versus how it behaves in plants. I would like to conduct an experiment to see if the proline-rich protein in humans is the same one in plants. I can use a biochemical approach to analyze the function of the proline-rich protein in a test tube. These types of investigations require a sample of the protein to perform enzymatic or binding assays to investigate the protein's activity. Thank you.